Alrighty guys, so we're going to talk about various pieces of evidence of the common ancestry of all organisms, how we determine relationships between different organisms, and also basically some of the basic pieces of evidence for evolution by natural selection. So here we go. The biggest, well, not the biggest anymore, but the foundational piece for us trying to piece together the relationships between different organisms, especially ones that are no longer in existence, so the extinct ones, is mostly based on the fossil record. And so we're going to take a look first at how the fossil record reveals kind of these broad patterns and how life evolves. For example, these guys are pretty famous fossilized organisms. They are totally extinct, have been for a couple 100 million years, but they're trilobites, and they're so common in the fossil record that a lot of times they were used as kind of an index, a baseline, like, oh hey, this other fossil was found in a layer above the trilobites, so we can guess an approximate age. So the basis of all of the knowledge we have of the fossil record rests on a few assumptions. One, the lower rock layers are usually older, and the higher or closer to the surface rock layers are usually younger. So when an organism dies and it ends up getting trapped in soil, gets covered in dirt, eventually more dirt, more sediment, more of that builds up on top of the organism and eventually gets compressed into rock, and that forms sedimentary rock. Well, as more sedimentary rock forms on top of older sedimentary rock, it can trap new fossils. And these fossils can take multiple forms. So the most common fossil that we tend to think of are the skeletal remains that we see in museums and such, but you can also have molds that show the impression of soft body structures. So some of the knowledge that we have of kind of more 3D shapes of some of these organisms are when they fossilized in such a way that it left a mold around their body. There are also trace fossils, things like footprints or impressions, that can also give us other clues. So, for example, there's a very famous set of fossilized footprints at Laetoli in Africa that are thought to have been made by a species of hominid, but not one in the genus Homo like we are. Instead, it is found in Australopithecus afarensis is the species thought to have made it, and it's very clear from the footprints that that organism is walking upright. So it starts to kind of help us see, okay, this is where we know at least this far back in our lineage we were walking upright, we were bipedal. So those can give us some clues. And the fossil record can do a really good job of kind of helping us figure out where some traits arose and where some traits kind of disappeared. So there are some traits that are ancestral, meaning that they were found in an ancestral species. So for example, we're looking at a fossil record showing the origin of tetrapods. Tetrapods are all the organisms that live on land and have 4 limbs. Includes us, includes all mammals, birds, reptiles, amphibians, and they are thought to have evolved from a water-living or at least semi-aquatic organism, and ultimately as far back as ray-finned fish. The ancestral traits that we share are, for example, our skeletal arrangement. And so we have this similar pattern of 1 upper arm bone or upper limb bone, 2 lower limb bones, and then a whole mess of smaller bones, which in humans take the form of like your carpals, metacarpals, and your phalanges. And you see that pattern repeated in all of these various species. So Eusthenopteron has that same pattern, which you can see in the fossils. Tiktaalik has that same pattern that you can see in the fossils. But around the time of Tiktaalik, you start to see an organism that has a limb that could actually support at least some of its weight on land. So it's thought that Tiktaalik at least went out on land somewhat because of the structure of its limbs. Then Acanthostega, Ichthyostega, all of those started developing something more similar to what we think of as the modern arrangement of limbs, in which case, some of them had more than the 5 fingers we tend to have on a hand. But basically, you see this kind of developing of these derived traits very similar to what we see in modern organisms. And it also helps you see where these lines split. So, for example, Eustheropteron, brain fart, is around 400 million years old. So that kind of split going more towards what was the ancestor of tetrapods happened then, whereas kind of the split between living tetrapods and one of, our extinct relatives like Tularpiton or Tularpiton, I don't know how to pronounce that one, was about 360 million years ago. So looks like a lot of this evolution happened in less than like 50 million years. How do we know, though, how old these fossils are, speaking of age? Well, there are two major ways that we can tell the age of a fossil: relative dating and absolute dating. And we're only going to talk about a specific example of one of each. So relative dating, like its name says, tells the age relative to something else. Is it older or is it younger than a layer of rock, than another fossil? And that's based on a lot of times stratigraphy, which is showing, which is looking at the order in which these rock layers are found. So the oldest layers are at the bottom, the youngest layers are at the top. Anything that's above another layer is going to be newer than it. Anything that's below another layer is going to be older than it. So these trilobites would be older than these clams because they're found in a lower level, a lower stratum or layer. Then there's absolute dating, which would give us an actual age in like numbers of years. And a very common example of that is radiometric dating, which looks at how radioactive isotopes decay in order to help determine the age. So if we look at stratigraphy in a little more detail, we'll see that we've got a couple of rules. So we always assume that the younger layers of rock are on top of the older layers. Everything was originally laid down horizontally. So if there's any tilting that happens, that would have had to have happened after the rock layers formed. Number 3 is not as important for us, but the law of lateral continuity is showing that, as long as you don't have anything else intruding in it or interrupting it, everything on the same horizontal layer is assumed to be deposited at the same time and therefore the same age. So if we had a fossil right here, we could then assume that it is the same age as a fossil in the same layer somewhere else. So that helps us kind of figure out relative ages. However, if we want to get a little more specific with it, we can look at the decay of some radioactive isotopes. You may be familiar with the term carbon-14 dating. Unfortunately, carbon-14 dating has a time limit on how long it's effective, but let's look at it for the basics of how this works. So a radioisotope is a radioactive isotope of a common element. So carbon-12 is the normal element that makes up most of the carbon in our body, but we all have a specific small proportion of the carbon in our body is carbon-14. Now, carbon-14 is radioactive and will break down into nitrogen over time. But, any carbon-14 that breaks down in your body, not going to really affect the long-term percentages of carbon-14 in your body because you're constantly taking in new carbon and compounds. However, once an organism dies, it stops taking in more carbon, and so we assume that all organisms have that same percentage of carbon-14 in their bodies, and so once they died, we know this percentage of their body mass should have had carbon-14. If we only find half of what we expected, then that means that half of that carbon-14 sample has decayed. And since all of these radioactive elements have a concept called a half-life, which is a specific time during which half of the atoms will decay and change into another element, and it's very reliable for a specific element. So the half-life for carbon-14 is around 5,700 years. So if you only have half the remaining carbon in a sample, then it's been about 5,700 years since that organism died. If you have only a 16th of the expected carbon remaining, then that means that you've been through 1, 2, 3, 4 half-lives, which means you've been through now 4 times 5,700 years. And so the amount of the isotope that's remaining lets you calculate how many years have passed. Now that is not going to be useful for carbon-14 for anything that's over about 60,000 years old. There's just too little carbon left by that point. However, there are some other elements, so uranium and potassium, that have longer half-lives that can possibly be used to date materials. Unfortunately, uranium is not found in living organisms, so you have to rely on finding uranium in a stratum like very close to that fossil that has uranium in it, so you can try and do that same detection. The fossil record is kind of a hit or miss sort of thing. So there's a lot of limitations in the fossil record. It's fragmentary for a lot of groups because not every organism fossilizes well. So for example, jellyfish are very, very rare fossils because they have only soft body parts, and it's very likely that they will decompose or be eaten before they get a chance to fossilize. So out of all the organisms that have ever existed on the planet, a tiny fraction of them would become fossils, and out of those, only a tiny fraction of them could be then in a place where they would be found by paleontologists. So if you had like this marine community, what you see on the image, that could be like what actually existed, the type of biodiversity you had there. But if you were judging based only on the fossil remains that you found, you might view it as only having these species because these are the only ones that fossilize. So we have to take that limitation into account when we are looking at like fossils and trying to judge what the ecosystems were like. In order to fossilize an organism, it's helpful if it's in a place where it's buried quickly by sediment or rock. And especially if it's in a low oxygen environment so that it cannot decompose very quickly, and also if it's in a place where it's hard for predators or scavengers to get to. And then there's also further gaps in the fossil record caused by the fact that, the rock cycle, rock moves around, sedimentary rock can get destroyed, and that will destroy any fossils that are in them. Other layers of sedimentary rock may be just too deep for us to, sorry, cat issues, for us to access them easily. However, the fossils we find do redefine our knowledge of the relationships between different organisms. So this is probably your standard mental image of a T-Rex. However, using new evidence and also using some fossils that were discovered in like the late 1800s, what we started to realize is that many dinosaurs had feathers. So this fossil is of Archaeopteryx, and it is a very important fossil. It's very famous, and what you can see is kind of the outline of feathers on wings. And then you see the outline of feathers around a tail. So scientists were able to kind of realize some of the other features of it besides its skeleton, and this is an artist's depiction of what Archaeopteryx may have looked like. It's hard to figure out color and things like that unless you're able to get some like pigment cells or something like that have been preserved. So the color is probably speculation, but based on this and other fossils, many of various dinosaurs that did have feathers, we now know that the closest living relatives of dinosaurs are actually birds. In fact, you can talk about the non-avian dinosaurs, or the non-bird ones, which are the ones that went extinct about 65 million years ago. And then there are the avian dinosaurs. So birds are little dinosaurs, which means, yes, sometimes evolution sucks. While a T-Rex is not necessarily the ancestor of a chicken, birds and therefore chickens are their closest living relatives, so yeah, kind of sucked for them. But hey, at least they're alive. Biogeography is also used as some evidence for evolution, and it fits very closely in with plate tectonic theory. So biogeography looks at the physical distribution of organisms. Where were they found? If we look in the fossil record and we look at where fossils of certain species were found, we'll be able to kind of, it helps us piece together what continents may have once been very closely and physically connected. So for example, back in the day when Pangaea was the supercontinent that contained all modern land masses, South America and Africa fit together almost like puzzle pieces. And this is substantiated by the fact that you'll see some fossil ranges that basically cross the boundaries of what are our current continents. So for example, this guy, the fern Glossopteris, is found in South America, Africa, the tip of India, Antarctica, as well as Australia, showing this kind of continuous range of habitat and showing that, okay, so those continents were once connected. Then if you look through the fossil record to the time right after Pangea, you'll see some of these species that were very similar now starting to pick up differences. And the farther you get from the spreading of Pangea, the more differences these different species accumulate until they start to develop into very distinct species. So the evidence all kind of ties together. And then now let's go away from the fossil record a little and look at something that where we can look at this with also some modern species. So we can look at morphological homology. Morphology is the study of the shape, the size, etc. of body parts. And so morphological homology is looking at the similarities and differences between body parts and different organisms. Unfortunately, for the species that are extinct, we mostly just have morphology to rely on, and that's formed a lot of our basis of our understanding of how species are related to each other, because that's what we have. We have their physical remains. There are some limitations, though. Some taxa may look very similar morphologically, but may have technically been different species because they were incapable of interbreeding. And then there's also some morphological variation that happens in the environment. And there's even like what's called gender dimorphism. So for example, in a lot of primate species, the males are much bigger than the females. And somebody who was just had no knowledge of primates and was just looking at the remains might think, oh, these two skeletal sets are different enough that we're going to consider them different species. And then also you have to look at, okay, what does the juvenile of the species look like? How can we tell that this is like a baby tyrannosaur instead of some completely different species? So there's a lot of stuff going on there you got to keep in mind. Some other pieces of evidence for evolution and common ancestry are homologous and vestigial structures. So homologous structures are similar. Remember, same location. They'll be in similar locations in the body. They'll have similar shapes and arrangement of structures. So, for example, here is Eusthenopteron, fishy ancestor of all tetrapods. Its forelimb, its like forefin, I guess, had this arrangement of bones that we see very commonly in the arms and legs of different organisms. It's got that one humerus bone, radius ulna, and then those carpal metacarpal phalanges. And if we look at all the organisms that are descended from that common ancestor, they share this same structure. They share that same one bone, two bone, lot of little bones. They've just been adapted to fit their current function. But you can see that evidence of common ancestry in those common traits. Then vestigial structures also show relatedness, but instead of showing relatedness for things that you have, they're more like body parts that are getting lost. So there are a number of vestigial structures in humans. For example, wisdom teeth, which when we had bigger jaws way back in the day, and I'm talking like, early Homo sapiens, so like 1 million years ago, then those wisdom teeth would come in after, about 15 years of age, when you might have lost some teeth already and give you some more space to chew. Now those are actually disappearing. There are people who are born with less than 4 wisdom teeth or who are born with none. And so those are a trait that may disappear eventually, especially since it's a disadvantage to have all your wisdom teeth because they get infected. Then the appendix is also vestigial. Its function was most likely in an ancestor related to digesting grasses and tough plant materials, and as our diet moved away from that, we started cooking things and finding fruits and things like that, then our appendix was smaller and smaller. Same thing with the tailbone. So primates don't have tails. What ended up happening is it must have become a disadvantage to have tails. So any organisms in the species who had long tails probably didn't survive, and the ones with shorter tails probably had more offspring. So gradually over time, in the population, you'd see that trait of tail size getting smaller and smaller and smaller until eventually you were left with primates who have no tails sticking out. Instead, they have that shortened version, which is a tailbone. You can also see some signals from development. So for example, all vertebrates have a structure called a notochord, very closely associated with your backbone, but not quite the same thing. That's the definition of being a vertebrate, is you have that notochord. If you look in adult organisms, sometimes it's very obvious. If you've ever dissected a frog, it's got a very obvious spinal cord. But in other species, like this guy, which is a sea squirt, you don't really see an obvious evidence of that. That does not look like a vertebrate. It looks kind of like a sponge. But if you look at the larvae of a sea squirt, you'll see that they actually have a notochord, which lets you know, oh, hey, this guy is a vertebrate. So it's probably more closely related to us than it is to sponges. And then you can also look at comparative embryology. So if you look at the embryos of different species at different points in development, you'll see that the more closely related species are, the longer they will look similar in the womb. So for example, these four are all mammals. And since they're mammals, they share a more recent common ancestor with each other than they do with any of these other organisms: fish, salamander, tortoise, chick, etc. Another thing, tortoises and chicks, that's going to be reptile and reptile descendants, which are, birds, which are some type of dinosaur. And notice that they look very similar for longer in their development. And so that's kind of also a mark of common ancestry. Another thing is, you see these traits that we have in early embryogenesis. We all have gill slits when we're little mini tiny little things, like the size of a shrimp. And we all have tails. So we all actually had a tail and gill slits at some point. Then you can look at molecular homology, and this is probably the biggest field, because we're just discovering so much with DNA and protein sequences, etc. And we can look at not only the DNA in the nucleus, we can also look at DNA in chloroplasts and mitochondria as well. We can also look at the amino acid sequences of mature proteins, and to be able to see, okay, what commonalities are there. So let's say that this is representing a DNA sequence or a protein sequence. And what you see is you have this for multiple species. You got a human, a mouse, a horse, a chicken, a fish, a snake, and a fruit fly. And what you'll see is that if this is a protein that has similar function in all of these organisms, and is something that doesn't mutate very quickly because it's so essential, then you might see small mutations build up. But the assumption is that all of these organisms inherited this gene from a common ancestor. Then, as their diverse species split and diverged, each of them started building up some mutations. Now, mutations didn't happen dramatically because this is an essential protein or an essential gene, and so you don't have a lot of room for having problems or mistakes, because once you start accumulating a lot of mutations, then there's a chance that you can have a harmful one that kills the organism. But you'll notice the three most closely related species on here, which are the human, the mouse, and the horse, were all mammals, have the most similarities in their sequences. Then you have chickens, which are slightly different, and they have a bit of a difference here. It's mostly in this region. Fish, which are even more distantly related to us, are going to have even more differences. Snakes are actually probably more closely, well, actually, not 100% sure. I think, yeah, they're more closely related to us than to fish, because they're also, they come from a tetrapod ancestor. But, it's not going to be 100% match of lineage and homology for all genes. So then we can look at the sequences of either nucleotides or of amino acids in a protein, and we can look at their similarities to see how closely related two species are, or more than one species are. So here we're looking at a sequence of a histone protein in a human, a chimp, a mouse, a rat, and a cow. And you'll notice that there are these segments where you have the same exact amino acids in the same location in each of these different species. Those are called conserved regions, and the more of these conserved regions that you have, the closer the evolutionary relationship between two species, because the thought is they've had less time to accumulate mutations between them. You may also see a diagram kind of like, or a set of data kind of like what you see on the right, where they're saying, what are the percent of amino acids that are identical in the same location as those of a human. So humans, obviously, we're 100% identical with humans. We all have very similar hemoglobin sequences. Rhesus monkeys share 95% of the sequence of their hemoglobin protein with us. So then you look at other species, and a lamprey, for example, only shares 14% of its DNA with us. So you can look at this data and say, okay, humans are much more closely related to rhesus monkeys than they are to lampreys. And the really cool thing is, in the past decade or so, we've actually been able to look at the DNA of extinct species and compare them with living organisms. So some of the organisms that we've sequenced the genomes for are Neanderthals and Denisovans, or Denisovans. Sorry, I mispronounce that all the time. Those are both two species of hominids that are closely related to ours. So closely related, in fact, that once we sequence those genomes, we're able to look at human genomes and find out that there was evidence of interbreeding between Neanderthals, Denisovans, and humans. In fact, if you are from anywhere other than sub-Saharan Africa, you've got a pretty good chance of having anywhere between 1 and 3% of your DNA in common with the Neanderthal genome. So some of the genes from Neanderthals made their way into the human population through interbreeding. So we're sequencing more and more genomes of extinct species. You may hear them talking about, like, oh, we sequenced mammoth DNA and stuff like that on the news. And that lets us look at kind of how closely related these were to modern species, as well as how closely related they were to each other. Because, for example, in samples like 4 or 5 mammoths, they were able to tell that there were actually 2 different species of mammoth represented in that sample. And then when you trace all of this back, look at all of the evidence, especially molecular homology, you can trace all of our lineage back to something called LUCA, the last universal common ancestor. So it's kind of evidence that we all ultimately evolved from some single-celled creature that probably resembled bacteria and archaea more than it resembled eukaryotes, and all lineages of everything on the planet trace back to this one last universal common ancestor. So, since all organisms share common ancestry, let's look at some of the pieces of evidence of this. So in all organisms, we all have our genetic information go from DNA to RNA to a protein sequence to a trait. So we all follow that central dogma of genetics. There are no exceptions to that in living organisms. The only thing that's like an exception are retroviruses, and we don't technically consider them alive. With few exceptions, all organisms use this same code for translating RNA to protein. So they all use basically the equivalent of this codon chart. The only exceptions are there are a few bacterial species that take this stop codon and actually use it for tryptophan instead. That's it. Everything else is absolutely identical for all organisms, which means this had to be the same for our last universal common ancestor, so LUCA had to use this same translation chart. Also, we have similar biomolecules that we use. So we all use the same 5 nitrogen bases, adenine, guanine, cytosine, uracil, and thymine in our DNA and our RNA. And then, if you look at this diagram, it says in the set of all amino acids, there's a small group of those that are what we call L isomers. Out of those, there's like a small section that have amines on an alpha carbon and all of these different characteristics. Well, out of this giant pool of possible amino acids, we only use 20 or 22. That's it. And all organisms only use those. So you don't need to know what it is, but a D-stereoisomer, no living creature uses these. Nobody. Even though they should function just as well. So obviously the last universal common ancestor used the same amino acids, same 20, and the same 5 nitrogen bases. There are also similarities in metabolic pathways, although part of this is going to be due to the fact that, for example, plants, their ancestor took in cyanobacteria, and then eventually those cyanobacteria that weren't digested became part of the plant cell as a chloroplast, and then eukaryotes took in mitochondria. They took in a bacterial species, and with endosymbiosis over time, they developed a relationship to where the mitochondria were the descendants of those ancient bacteria. But we still use very similar mechanisms to get energy. Things like NADP or FAD or NAD, all of those kind of electron carriers and hydrogen pumps and ATP synthase are all common mechanisms. And then what you see here on the left, those are some of the molecules involved in photosynthesis that are found in the thylakoid membranes, either in cyanobacteria or in plants, and they're very structurally similar. Speaking of common ancestry, all eukaryotes also have a common ancestor. Sometime after Luca, probably about 1 billion years or so, the first eukaryotes arose, and we all share a common ancestor. You can see this because we all have similar structures. We all have, for example, we all have mitochondria. There's it in the animal cell, plant cell, and a fungal cell. We all have nuclei. We all have rough ER, and I could keep going through the organelles, but you see the trend. We all have these similar organelles that are performing similar functions with similar structures. We just have some that are kind of modified. Another similarity is we all have multiple chromosomes, and they're all linear chromosomes. So here we see an actual microscope image of plant chromosomes. Notice that they're linear, single long line. I could not find a good microscope image of fungal chromosomes for some reason, but I was able to find this as like a diagram showing the various chromosomes in a yeast. And then here are some chromosomes from a number of animal cells. So you just need to know that they're linear and there's more than one of them. Not really anything else about that diagram. All of our genes have introns. We have at least some genes that have introns in them, and all of those introns are removed by a spliceosome. And that spliceosome has like 5 small nuclear RNAs and a bunch of proteins put together. So we all have introns that we splice out. And that's it. So now that we've looked at the evidence of common ancestry, we're going to look at how we diagram it next. And that's going to be when we look at phylogeny in our next lesson.